early and precipitous start. First speaker in the session on novel antibody technology is Kenneth Shea from University of California, Irvine, speaking on plastic antibodies, synthetic receptors for biological molecules. He doesn't say whether it's polystyrene or PVC.
Thank you very much, and I want to begin by thanking Aubrey for his very kind invitation to participate in this symposium. My talk will be quite a bit different from synthetic chemistry, and I will work directly on the important problems that you are involved with. But we work on developing new tools that might be of value on the projects that you're working on. So I'll begin by giving a bit of background. About 5.5 years ago, we challenged with the design, synthesis, and evaluation of synthetic materials that might function as an antibody. As the title suggests, we're trying to develop synthetic polymers. These are abiotic.
Thank you. These will be synthetic polymers that is completely abiotic that can perform with the affinity and selectivity of an antibody. That was the challenge that we posed ourselves. Capture agents for peptides and proteins as I've illustrated on the slide. So this is the challenge. General strategy for preparing synthetic polymers with antibody-like affinity and selectivity for peptides and proteins. Now, just to make this clear, there are a number of nanoparticles that are used as delivery vehicles for diagnostics and for delivery of therapeutics. Typically, these nanoparticles are functionalized with ligands targeting a particular receptor at the intended site. They also might be decorated with antibodies, again, to direct them to a particular site in a living system. We're talking about the design of a synthetic polymer with an intrinsic affinity for a particular biological macromolecule, as illustrated in this particular case. So a polymer with an intrinsic affinity for biomacromolecules. Synthetic chemists over the years have really refined skills in crafting small molecules that are capable of entering a biological system, seeking and finding a specific protein target, binding to it and influencing or mediating its activity. Okay? This is the cornerstone of the current pharmaceutical industry. Design a small molecule for a well-defined, or reasonably well-defined binding site, and people have developed those exquisite matchups like this. The problem that we're confronting is a bit more complicated. We're not targeting a specific well-defined site. We're in fact targeting a biological macromolecule, a peptide or a protein. And how to go about designing and evaluating a synthetic entity that can recognize a complex entity such as this requires a somewhat different strategy. One of the insights that we take is from protein-protein bonding itself. If we look a little bit more carefully at protein-protein interactions, those interaction sites expand over hundreds of square angstroms. Okay? And if we look a little more closely at a higher level of resolution, these interactions that take place on a protein-protein complex involve individually weak but collectively strong interactions, hydrogen bonding, electrostatic, van der Waals, and things of that sort. Okay? And so the lesson that we take from this particular system here, protein-protein interactions, is the fact that it's hundreds of square angstroms, collectively, individually weak interactions. Can we craft a synthetic entity that has a presentation of functional groups that's complementary to the intended target? Now, we're doing synthetic polymer chemistry. We cannot control the sequence of the functional groups on the polymer chain, but we're relying upon averages of functionality that would be complementary to the intended target. Remember, these interactions presumably are occurring over hundreds of square angstroms. We also want to introduce an element of flexibility into this polymer chain so that these functional groups can adapt, okay, to the presentation of functional groups on the protein or biomacromolecule target. So that's the concept that we have come up with in order to create synthetic polymers that might have a chance of recognizing a specific domain of a biological macromolecule. Now, to place this in context, there are many approaches to create synthetic receptors for proteins and peptides, and Mike will be talking about one of the approaches, aptamers, I think, or aptamers in one of the following talks. So these are the approaches that are currently being used, some with very promising success. Aptamers, phage display for peptides. All of these can be screened against the intended biological macromolecule to find an entity capable of binding selectively and with high affinity to a particular target. Our approach is a bit different. As I indicated, for a synthetic polymer nanoparticle, we focus on two aspects. The first is a crafting of this synthetic entity with complementary functional groups to the intended domain of the biological macromolecule. The second part is to use a technique called molecular imprinting in order to heighten the affinity and selectivity of the synthetic polymer. Molecular imprinting consists of adding a small amount of, for example, an epitope of the intended target into the polymerization reaction with the hope of improving both selectivity and affinity. A cartoon and highly idealized representation of this is shown on this slide. If this yellow tennis ball is the intended target, what we do is mix it with polymerizable functional monomers. These functional groups are complementary to sites or domains on the intended target, and this is a cross-linking entity. The polymerization takes place, and the imprint molecule is used to organize the functional groups to create in 3 dimensions a binding site. Following polymerization, this template or imprint molecule has to be removed. This is a highly idealized representation of what really looks like a rat's nest, okay? And we'll have more to say about that shortly. But complementary functional groups are included in the polymerization reaction together with, at the end of the day, okay, a small amount of the epitope of the intended target. I'll first give several examples which got us excited about this. It is the molecular imprinting part, and then I'm going to go where we combine both strategies to create high-affinity nanoparticles for biological macromolecules. So in our initial quest to see if we could create a synthetic polymer capable of recognizing a complex protein, we identified an unstructured domain that is exposed, an epitope, the same strategy, of course, that people that are developing antibodies use. The question of how big an epitope and its composition, well, we worked with a group in bioinformatics to help us with this, but we chose a nanoparticle-peptide. This is a unique sequence to this particular protein. The strategy then, if this is the protein that we want to target and capture, we identify an epitope, we synthesize it, we carry out our polymerization. This is the imprinting step where a small amount of the epitope is included in the polymerization reaction. Following polymerization, a material is formed. We then dialyze or remove the epitope to produce a synthetic polymer, a network polymer, and we then interrogate it to see whether it's capable of actually capturing the intended protein target. First example that we did in this system was to a capture agent for cytochrome C. We interrogated surface and identified several potential epitopes to use. In this particular case, we chose the C-terminus epitope, the exposed, unstructured C-terminus epitope of cytochrome C. This is the peptide sequence of that C-terminus, and so the next step is to synthesize this particular peptide. What we're going to be doing now is taking that epitope and creating on top of it a synthetic polymer film. Once that film has been crafted, we're going to take the film and add it to a solution of 5 different proteins, incubate it, and then remove it, extract it, and then evaluate whether in fact we have selectively pulled out the intended protein target. And so the process is illustrated as shown here. This is the C-terminus epitope of cytochrome C. It's covalently attached to the surface of a glass slide by amide coupling chemistry. Polymerizable, water-soluble monomers are then poured on top of this glass slide. We use a photochemical initiator to produce a polymer film on top of the glass slide. We next then incubate this The film separates by gently swelling, and then we take that polymer film and add it to a solution of 5 different proteins. Once equilibration is achieved, we take the film out, wash it gently to remove that which is only superficially bound, and then we do a serious extraction to see whether it has a high affinity for one of the proteins. And in this particular case, this is a page display, SDS page display. These are the 5 proteins that were present in the mixture that we incubated our piece of polymer film, and this is what it pulled out, the imprinted polymer. A good deal, although this is a sloppy page gel, cytochrome C. A control polymer consisting of the same monomers, but made on a surface that did not contain the epitope, is shown in the far lane over here. Well, this caused a great deal of excitement in the group, but also it might have just been a lucky stroke. Proteins are complicated substances. We have a polymer film. Maybe cytochrome C is simply sticky. And so to give any validity to this sort of thing, or any notion of generality, I should point out that the affinity of cytochrome C for that film is in the nanomolar regime. But to see whether this method is general or just a fluke for this particular protein, what we did is go through the entire process again, only this time a different protein target with a different epitope sequence is shown here. This is BSA, this is its C-terminus. It was attached to a glass film and a glass slide. A new polymer film was made, taken through the same process and incubated with those same 5 proteins. And this is what this polymer film put out. Not cytochrome C, but BSA. This lent a little bit more credibility then to the generality of it. And we've done this 6 or 7 times. This is one where we've used alcohol dehydrogenase. Once again, using a completely different epitope, okay, as our imprint molecule to produce a synthetic polymer capable of pulling out some alcohol dehydrogenase, not cytochrome C or BSA, which was pulled out by this polymer film when it was made in the presence of their unique epitopes. We've actually established it early on in this case to show that this technique has sensitivity in some cases to the single amino acid residue. So the native BSA is capable of pulling out BSA protein, but if we imprint with epitopes that contain a single amino acid change in the center, in fact, the affinity for the protein falls off. This was all quite exciting, but we also appreciated that the film technology is a relatively low capacity technology. That is, the amount of protein that might be pulled out by these polymer films, since it's a surface phenomena, is quite small. And if we were going to explore a higher capacity medium and one with greater opportunities for applications, we would have to go to higher surface area media, such as a nanoparticle. The challenge here is to develop chemical methods to synthesize our polymer in the presence of an epitope, okay, and yet produce reasonably uniform nanoparticles. We've developed several different polymerization methods, microemulsion polymerization and precipitation polymerization. These may not mean much, but they give us an opportunity to prepare nanoparticles with selectivity for particular proteins and peptides over a range of peptide solubilities, if that's what we're using for epitopes, from water-soluble to more hydrophobic ones. Now I'm going to transition to the general approach that we've developed since these initial experiments in confirming to ourselves, at least, that one is capable by polymerizing in the presence of a small amount of epitope to produce a synthetic network polymer capable of selectively capturing a complex protein. The process now is integrated with functional group optimization, and in fact, this is where we spend most of our time. So what we're going to be doing is, given an intended or target peptide or protein, we're going to be developing polymerization recipes containing a variety of monomers that produce an intrinsic affinity for that particular biomacromolecule. Okay, so optimize the monomer composition. We're talking about functionality, complementary functionality to the intended biotarget. Also, in consideration of potential therapeutic applications, we have to be concerned about the specificity, that is, common proteins that are encountered in the serum, We want to minimize those types of interactions, and once we think we have the right formulation, we then want to add a bit of the epitope, the final step, to see if we can improve both affinity and selectivity. This is the protocol that we're now following for synthesizing synthetic polymer nanoparticles, okay, with reasonably high affinity and selectivity for target biomacromolecules. The example that I'll talk a bit about is our work with the peptide Melittin. Okay, it's a 26 amino acid peptide. It's the principal component of honeybee venom. It's an amphiphilic peptide. It's a pore-forming toxin. Okay, and we take advantage of that property a little bit later, as I'll show you. So if we were to design a synthetic polymer nanoparticle that had a high affinity and selectivity for this particular peptide, we evaluate what its functional group presentation is. In this particular case, the brown here represents relatively hydrophobic domain. The blue here represents positively charged amino acid residues. So this is like a bit of a detergent molecule, both hydrophobic and charged. And in consideration of that, we take from our library of readily available polymerizable functional monomers functionality, positive charge, hydrophobic, negative charge, hydrogen bonding, and what have you. These are the monomers that we mix and match in our initial formulation to produce nanoparticles. We polymerize it using this precipitation polymerization. This is an aqueous-based polymerization, so we start with a homogeneous solution of monomers, a free radical initiator, and a small amount of detergent, and that recipe produces nanoparticles, reasonably uniform, typically within the size range of 30 to 50 nanometers. Those nanometers are hydrogels, relatively lightly cross-linked, okay, containing various amounts of the monomers that we include in the polymerization reaction itself. This is a composition of a small library of nanoparticles that are made using this recipe. The color codes corresponds to the functionality and the amount in the individual nanoparticles. As I indicated, the nanoparticles are evaluated after extensive dialysis, I should say, by AFM and dynamic light scattering. These are lightly cross-linked hydrogel polymers, and isopropyl acrylamide, NiPAM, is the dominant monomer in this particular case. The next step of this is to evaluate whether these individual nanoparticles have any affinity, any intrinsic affinity for the melittin peptide itself. And one of the techniques that we use is a quartz crystal microbalance. Some of you may be familiar with this, perhaps all of you, but essentially it's an oscillating quartz crystal, and it's an extraordinarily sensitive analytical balance. It has a gold surface, and so we can use standard gold coupling technologies to attach stuff to that surface. In this particular case, on the quartz crystal microbalance surface, we attach melittin. And what we do is flow through the quartz crystal microbalance cell solutions of our libraries of nanoparticles. If the nanoparticles show no interaction with the melittin on the surface, there's no change in the frequency of that quartz crystal microbalance. Those nanoparticles that sit down or have an affinity for the melittin on the surface will change the frequency. This is the frequency change of nanoparticles that have little or no interaction with the surface. These are frequency changes that take place upon addition of nanoparticles that have an affinity for the melittin on the surface. We use this technique and other protocols to evaluate whether our nanoparticles have any affinity for melittin. And from these studies, what we have learned is that many of the nanoparticles, this is the change in frequency, have no affinity for the particular melittin that sits on the surface, but we do have several hits, okay? And what is the composition of those nanoparticles? Well, perhaps not surprisingly, these are NiPAM-based, but the hits that we observe, the nanoparticles with an intrinsic affinity for melittin, are comprised of hydrophobic domains and carboxylic acid, negative charge. Remember, melittin is a hydrophobic, positively charged peptide. So these are the leads that we take into a subsequent optimization protocol. We also use techniques to evaluate the affinity of our nanoparticles using a red blood cell test. Remember, I indicated melittin, its mode of action lyses cells. And so we use the red blood cell test and the ability of a nanoparticle to suppress melittin's ability to lyse red blood cells as one of the diagnostics. So this is a tube containing red blood cells that have been centrifuged down. Next tube contains the same red blood cells to which melittin has been added and then centrifuged. The lysed cells release hemoglobin, red in the supernatant, and the color in the supernatant tells us the degree of lysed cells in this particular case. Subsequent tubes here, we have added nanoparticles from our small library. Most have no effect at all. Some seem to enhance the red blood cell lysing, but this tube here, in fact, seems to have suppressed melittin's ability to lyse red blood cells. And what are those? Well, once again, and this was interesting to us, it required a mixture of functional monomers, hydrophobic and negatively charged. So nanoparticles that were composed of just negative charge to rely upon electrostatic interaction, or those that just had hydrophobic domains, were ineffective in capturing it. And we needed both of these monomers in rather well-defined ratios to have a nanoparticle with a high intrinsic affinity. We then go back and optimize this, okay, to find what we think is the nanoparticle composition that has the highest intrinsic affinity for this particular biomacromolecule target. And this is a plot using the red blood cell test to evaluate the affinity. We have found then a composition of a nanoparticle, a chemical composition, that has a high intrinsic affinity for melittin itself. The last step in the process then is an imprinting step where we take those monomers that give us the high affinity nanoparticle and we add a small amount of melittin itself in the polymerization. And that we've learned gives us an added bump in both affinity and selectivity. Following polymerization, extensive dialysis to remove the melittin and any unreacted monomers or oligomers, and then we take these nanoparticles on for evaluation. We can evaluate the capacity of melittin that these nanoparticles have using sort of the neutralization of the red blood cell test, and we compare that capacity to an antibody that has an antibody for melittin. We're looking on a per weight basis of our synthetic polymer nanoparticle that has a comparable capacity to the nanoparticle, to the antibody, excuse me. Having developed then this nanoparticle with a high affinity and nanomolar affinity for melittin, we're interested to see whether it could actually perform in a complicated setting. And so we did this rather cautiously, beginning with in vitro studies, following first establishing that the nanoparticles themselves were not intrinsically toxic, cytotoxic. This allowed us to proceed with in vitro studies. Once we had successful results there, we then moved on to in vivo studies, and I'll tell you a little bit about those. After we established the absence of cytotoxicity of these polymer nanoparticles, we looked at survival rates of these fibrocarcinoma cells, and we observed that their survival rate, these are the ones that have been dosed with melittin, their survival rate was related to the amount of nanoparticles that we added to that mixture. Okay? And so we established that, first of all, the nanoparticles were not cytotoxic, and they were capable of performing in the in vitro study by inhibition of cytotoxicity of melittin itself. After a rather lengthy series of experiments, we then moved on to in vivo studies with mice, following first protocols to establish the absence of toxicity in mice, and then also to establish that there were no indications of immunogenic responses. These are after 2 weeks, a comparison with controls, with weight gain that mapped onto the control mice, and also then autopsies of these mice after 2 weeks to look for any signs of an immunogenic response, and there were none. This allowed us then to proceed to the next stage, which involved injection of a lethal dose of melittin into the mouse, followed on approximately 30 to 60 seconds by an injection of the nanoparticles itself. Thank you. I should point out any study with nanoparticles in complex biological systems is fraught with potential difficulties. Pristine nanoparticles become immediately coated with common serum proteins. and this can alter or completely suppress the intended function of the nanoparticle. We did, in fact, look at these data, and in fact, these nanoparticles showed little strong interactions with common serum proteins. The results of the in vivo study are shown here. The green curve, in fact, is the survival rate of mice that were subjected to a lethal dose of melittin. A subsequent dose, 30 seconds thereafter, of the nanoparticles then improved their survival rate, and a larger dose further improved that survival rate. I'll rush through this data, which is comparable. A smaller dose of melittin was injected into the mice, followed by an injection of nanoparticles, and that lethal dose, was suppressed. There was presumably a cause and effect relationship associated with these results. We'd like to think that the nanoparticle was involved somehow in neutralizing the melittin in the blood, And so in order to get additional information, both the melittin and the nanoparticle were fluorescently labeled. And this allowed us to track in real time with mice that had been injected just melittin, and then mice injected with nanoparticles, 2 different types of nanoparticles, so we could observe how the nanoparticles dramatically altered the distribution of the toxin in mice. Following these studies, we were able to do autopsy to establish where, in fact, the nanoparticles and the melittin wound up. They were concentrated, perhaps not surprisingly, in the liver, concentrated in the liver, and this is the slide, the 2 sections of liver tissue showing the melittin, which is associated with this dye, the green dye, and the nanoparticle, which is associated with the fluorescein dye, and the superimposition shows that both melittin and the nanoparticle are together in macrophages in mice, One last point, and I'll do this rather quickly. A reality check. We are making synthetic polymer nanoparticles. These are not pure substances. We have a distribution of sizes. There's also a distribution of affinities, So these are, at very best, poor polyclonals. But because they're nanometer in size, and because they have an affinity and selectivity for a particular peptide, we have an opportunity to purify them, to tighten the distribution both in size and affinity using an affinity selection technique. And here we take advantage of a rather interesting property associated with the polymer nanoparticles that we make. They exhibit an interesting temperature effect on their solubility. Counterintuitively, at higher temperatures, these are less soluble and they're collapsed. At lower temperatures, they swell. These are made and evaluated in mice at higher temperatures where they're collapsed, where they have high affinity. We've taken advantage of this by simply decorating an agarose bead with melittin. And then at room temperature, where these beads have high affinity, the synthetic polymer nanoparticles have high affinity, we then simply incubate them, and the high affinity nanoparticles will associate with the melittin. We then simply flush the column, removing most of the beads, but left behind are those beads with very high affinity. We simply cool the column down to 5 degrees. They expand, become highly swollen, no longer have affinity, and then we can collect nanomolar or subnanomolar affinity synthetic polymer nanoparticles for it. So I'll conclude by just pointing out several of the important potential advantages associated with these materials. It's a relatively short time from discovery production. They're abiotic. They do not require animals or live cells for their production. They can be readily integrated into existing assay formats, Currently, we're investigating a number of applications, and I'll finish by simply listing them shown here. These are the current research projects. And what's most important to acknowledge all of the students and collaborators that have been associated. The current group that is with me on this project at UCI are shown. These are former students. We've had ongoing collaborations with the group in bioanalytical chemistry, Okahata at Tokyo Institute of Technology. And the animal studies were done with Professor Oku at the University of Shizuoka. Primary support has come from NIH. And thank you very much for attending.
Thank you very much, Dr. Shea. We have time for perhaps one quick question.
Yes.
Please.
Have you considered making nanoparticles that bind lipopolysaccharide, the most toxic substance in the world that kills roughly 300 million people per year and costs $15 billion a year spent on intensive care units, and nevertheless 9 out of 10 of the patients die anyway, so that's not very well spent money.
It seems like an excellent suggestion. And yes, we have just completed a rather basic study using isothermal titration and calorimetry of our nanoparticles interacting first with carbohydrates. So we learned something about optimum compositions of that. And this target that you mentioned is, in fact, one of the ones that we're giving serious consideration to. Thank you.
Thank you very much, Dr. Shea.